Hi. Right now I'm going to explain to you what is this instrument. It's essential in any laboratory. If you're in biology, if you're in chemistry, you're doing research. So this equipment is called autoclave. And it's essential, like I mentioned before, because you have to use a lot of glassware. And the glassware has to be always sterilized since you're going to be working with bacteria, DNA, with proteins. So this is the only way that you can be sure that your glassware is going to be clean. So as you can see here, it has a temperature, and the highest temperature that this autoclave will reach is 121 centigrades. Remember, in the laboratory always we use centigrades. We don't talk about Fahrenheit, even though you are more used to use Fahrenheit. In the laboratory, everything is going to be centigrades. So here we have 121. In this autoclave, you can put scissors, any kind of silverware, like maybe for surgery, clothing, and glassware. So the purpose for this autoclave in this laboratory is because we put glassware. Right now, we just finished a cycle. So as you can see, this autoclave already finished the cycle. It's already cold. So I'm going to open it, and it's going to be like this. It's like if it was a safety valve. You have to be very, very careful. But one of the main things that you have to remember is that every time when you open an autoclave door, it doesn't matter what is the size, you always have to be behind the door, because the steam can come over and can burn you. So that's why this is at the right position. After I see that nothing is coming here, you will go inside, and then you start collecting your items that they were ready on the autoclave. So that's how you're going to be sure that this glassware is ready to be used in any kind of laboratory experiment. So then after you're done, you're going to close it again, and then you can reuse it, put new glassware ready for the next cycle. And remember, it has to be very, very tight. So no steam, nothing, no vapor. It's coming out. And when you cannot go any further, then you press start, and it will start the cycle again. And it will be like taking from between 30 to 45 minutes. And then the alarm will beep, and it will be ready for the next use. So this is the autoclave. I'm going to give you a tour about what is a biotechnology lab here at Silva, El Paso, Texas. And as you come in, you're going to see a lot of equipment and bottles and tools and toys. Well, we call them toys, but in reality, this is equipment that we need. So then, like this one, it's a shaking water bath. This one, what it has, you put water here, and then it can be moving in order to grow yeast, because the water has to have a certain temperature, but it has to be moving in order to increase the growth. We have incubators, which in this one is very special, because on this one, what you can do, this is the incubator. The temperature has to be at 37 degrees, because what is our body temperature? You can tell me, 37 degrees. So where do we put this one? This is going to be our Petri dish. You place it, remember, in the right position. The bottom goes up, then you close it, check that the temperature is at 37, and then you wait until 24 hours. The next day you come, you open, and you check for growth. And I have two of these. This is another shaker. On this one, what it's going to do, sometimes you have to have plates that they're going to be rotating. So you just, and this one, sometimes you have to put plates here, for staining, and you can adjust the movement, the velocity here for RPM, so it can go slower or faster. So this one will be a rocking platform. Then we have a PCR. This one, you have heard of PCR, for polymerase chain reaction. You heard about this one when we were studying the COVID, because here is where you can put the tubes here, and then they're going to go into different cycles, 3 cycles, and the DNA is going to start developing and breaking and growing. So after 3 hours, you will have billions and billions of DNA. What is this? This is a water bath. You put water here, and then you put your tubes here to float. And again, you can adjust it to the temperature that you need. It could be 55, 65, 37. So it depends how you need it. Then this is another style of a PCR. So we have another PCR. Here we have another water bath. And this one is a mini rocker, like the one that I showed you before, but this is a smaller one. Then we have the spectrophotometer here. And this one, what you do on this one, you're going to start reading absorbance. You put a cuvette here, and then it will go through the reading the absorbance of the light, and you will have a reading for experiments with the DNA or with protein or RNA. This is another PCR, and this one is a desiccator. So in this one, what you do when you have your electrophoresis gels, you will put it here, and it's going to start drying because of the material is made of a gelatin, and they will dry. And at the end, you can have this like kind of plastic, and you can cut it and put it on your notebooks, and you can see your DNA here. So you will have the lines. So that will be, useful when you have to make reports for research. Then, guess what? This laboratory is like a kitchen. So we have a microwave, right? It's the same as you have at home, but guess what? No food allowed. So we have this microwave here in the laboratory, but it's because we needed to start warming some reagents, liquid, water, some other media. So that's what is very helpful. That you open it, you put it here, for 30 seconds, close it, and then you have hot water. Okay, so now we have here the centrifuge, which is going to be one of the special tools that you need very daily, and for a lot of reasons. One is because if you want to mix reagents, then you're going to put tubes here. Remember, one of the rules is always you have to balance with the same amount of volume of the liquid, and one in front of the other one. Then you will close it. It will be like this, right? Few seconds, you wait. You have to wait until it stops. Then you open, and then you have either, sometimes you have like liquid on the surface, on the lid, and you want everything to go down. So the easiest way is to use your centrifuge. Okay, another instrument that we have here that we use very common is the vortex, and the reason on this one is sometimes you need it to shake, or you have sediment at the bottom or pellets, and you really want to them into the liquid. So the easy way is to use one of these, and you have to be gentle. You can adjust the speed. It can be slower, and then at the end, just go like that. And whatever is here that it needs to be fixed, mixed into the liquid, and to be equal, is going to be used after you use the vortex. So this is a vortex. One of the most familiar equipment that maybe you have seen before in your laboratory is going to be the microscope. That one is going to be a great tool for you because you're going to start looking into the bacteria, cells, everything that is tiny. When you're doing forensics, you can see hair, you can see skin if you're doing any kind of research. So this is going to be one of the best friends. So always you have to treat it with care. You have to know how to handle it, how to carry it, how to clean it, because these lenses are very fragile. And if you put your finger like that, it's going to be hard to get the oil from your finger. So never do that. This one has 4 objectives, and one is for oil. So if you don't know how to handle the microscope, it's better to ask instead of just messing up your microscope, because later it's going to be hard to put it to work and be fine again. So be careful.